Okay, so we're going to move on to the next talk. Actually, as you know, many of us think of David as a godfather, as a scientific godfather. But the next speaker, Leah Rosen, is actually David's goddaughter. Leah is a postdoc researcher in Stockholm, I understand, and she's a computational scientist, so she's going to tell us about our interaction with David.
Oh, my presenter notes are gone, so I am going to, wait, never mind. I'm going to have to wing it. So David has been amongst the handful of most important people in my life since before I can remember, before I knew what science was. I always wanted to be in his and Ros's presence. Their home was full of warmth, fun, adventure, and always the most interesting people. And luckily for me, it was right next door, and I was always welcome. When I was 14, I was living in Germany at the time, and I remember visiting Ros and David in London. On my first day, I got to visit Lincoln's Inn Field with David, and I remember looking down a microscope and seeing flies with legs where their antennae should be. Oh, I have my presenter view, great. It certainly left, and those flies with legs for antennae certainly left more of an impression than the museums dragged me around on Ros dragged me around on day 2. My passion for biology grew from there, and when I was 16, David arranged for me to spend 2 weeks in the RAF lab, in Jordan Raff's lab at the Dun School in Oxford. And if TV had sort of suggested to me that scientists were all alone, male, stuck in windowless labs all day, and villains, David couldn't have chosen a worse lab. David lived and therefore created a very different scientific reality. Naturally, he sent me to a lab where science was fun, social, I was mentored by a woman, and scientists were wonderful people who had labs with big windows and lunch and tea breaks outside every day. And every PCR always worked. And today I am a scientist. I went to university convinced I wanted to become a developmental geneticist, his words, just like David. Much to my mother's chagrin, especially when upon doing my PhD at the University of Cambridge, I joined his old college, Darwin, instead of hers. But while I was sidetracked by math, statistics, and computer science, my fascination with those flies with legs on their heads remained a through line. And in my PhD, I studied mouse gastrulation and early organogenesis with a particular interest in the epigenetics. Although by that point I had learned that PCRs don't in fact always work, and life was better as a computational biologist because I was just too clumsy in the lab. David's sincere interest in me and my science was a constant through line. I remember a car journey in Berlin with Ros, my mum, and David after I'd had quite a rough lab experience. David understood my feelings and convinced me that science didn't have to be the way I had just experienced it. This really rekindled my love for science, and I don't think I'd be a scientist today without that car ride. After that, whenever I saw Ros and David, Ros made sure to carve out time for me to talk to David about science, time when, oddly, she had other places to be. But David was always there. He always shared my excitement when things were going well and reassured me when things were tough. And even in February of last year, when he was already quite unwell, he, on the phone, deflected questions about how he was doing and instead talked to me for an hour about how my PhD thesis writing was going. And I received a thesis writing care package shortly thereafter. David was delighted when I told him that I was going to move to Stockholm for a postdoc. However, he sadly passed away before I was able to start. And if I can hold it together, I was never able to talk to him about the research I am now doing. And while Ros's enthusiasm for when I droned on about it to her certainly maybe even outstripped David's, David would have been a bit more critical. I do really, really wish David was still here so I could tell him what I'm working on now. And so in lieu of my godfather, I'm rather narcissistically talking to a whole room of people about it for just a few more minutes. It will mean a lot to me because I so wish I could tell him this. So when we study genetics, we're often interested in how a specific genetic variant affects phenotype and disease. And when I mean phenotype, I mean that very broadly in terms of cell state, tissue function, organismal phenotype, and disease. However, it's not just magically goes from genetic variant to phenotype. There's some molecular effect that bridges that gap. And we know that many of these genetic variants affect gene expression. For instance, here's an example. And GWAS studies have recently done a really good job at linking genetic variants to the phenotypes and diseases they cause. And we're also increasingly being able to link these genetic variants to the genes whose dosage they modulate in cis. However, the gap sort of between cis gene dosage and the phenotype and disease is modulated through sort of further things, which I'm calling trans effects. And when I say cis and trans, what I mean is that the genetic variant is in close proximity to the gene that it regulates in cis, and the change in the dosage of the cis gene then causes a trans effect, such as the change in the expression level of a trans gene. So recently, it's only been in the past few years that the scientific community has started studying gene dosage response. And when I say gene dosage response, what I mean is that you can have some dosage to trans effect. Here I'm calling it the function curve, like this. You can think about it in terms of homozygous and heterozygous. So if you have two functional copies of a gene, you're in the green range by the plus plus. Maybe I will. You're here. If you're heterozygous for the gene, you're here in the middle. And if you're homozygous negative, so you have no functional copies of the gene, you have none of the gene, so you're down here at 0. And I hope you can appreciate that recessive gene might be somewhere on this yellow line, where the heterozygote looks more like the homozygous negative, whereas a dominant gene will look more like on the green line, where the heterozygote looks more like the wild type. And we can continue these curves into overexpression. The dosage to function curve doesn't have to be positive, it could also be negative or non-monotonic. And so recently, we've been able to generate lots of data that we can use to try to infer these dosage to function curves, but they're often noisy in single cell data. And so in my postdoc, I've been developing a computational method, which I'm calling Bayes-Dream, that takes these very noisy single cell data and tries to infer the underlying dosage to function curve. And I felt like this talk wouldn't be complete without acknowledgements, but all I really want to say is thank you to my parents for impeccable taste in godparents, and a huge thank you to Ros and David for the profound impact that they've had on my life.
Thank you, Leah. I think David will have been very proud of your work. Any question? Well, probably not. All right, so we're moving on. Our next speaker is... Oh, there's a question. Sorry, I didn't see you.
I wanted to leave the stage to Ben.
I know, you have to come back.
Sorry, this might be a really stupid question, but so how do you deal with noise or variation between? Can you deal with that? So if you have gene expression variation between individual cells or organisms?
Yeah, so I'm currently working in K562, or well, cell lines, so it's quite non-physiological. There is a lot of noise, but I think we have statistical methods that are able to model that noise to some extent and fit means. I don't know how, I think there's all models are bad, some models are useful. So there's some level of complexity which we're not able to capture, but I think general trends we are able to capture. So I think we're definitely able to fit sort of 5-dimensional functions. Higher than that, I'm not sure yet.